Salut tout le monde, on va commencer tranquillement. Bienvenue à cette conférence de l'UIS. Aujourd'hui, on reçoit Hisao Moriya, professeur à l'Université de Koyama, au Japon. Il est professeur à la faculté de Environmental Life, Natural Science and Technology à cette université. Hisao fait sa formation en biologie moléculaire et a fait son doctorat à l'Université de Koyama, au Japon. Il a travaillé sur les éducateurs à ARN, chez plusieurs pays. Puis ensuite, il a fait deux postdocs. Un premier postdoc aux États-Unis, à l'Université de Washington à Saint-Louis, avec Mark Thompson, qui est un des pionniers de la recherche en génétique. Il a travaillé sur le métabolisme. Puis ensuite, il a fait un postdoc à Tokyo avec Hiroki Kitano, c'est le Systems Biology Tokyo, Systems Biology Institute de Tokyo, qui est un des premiers instituts en biologie des systèmes. Puis Kitano, c'est un des fondateurs du domaine de la biologie des systèmes. Au début, dans le domaine, il y avait des articles publiés dans Science. Pour nous expliquer c'était quoi la biologie des systèmes, c'est lui qui nous expliquait ça. Maintenant, c'est quelque chose qui est beaucoup plus répandu. Et à ce moment-là, il a travaillé sur la robustesse de l'expression des gènes pendant le cyclage, Systems Biology, avec un peu de modélisation. Et depuis plusieurs années, il dirige son groupe de recherche, travaille sur les contraintes qu'impose la cellule sur l'expression le niveau d'expression des protéines. Ils ont développé plein d'outils super intéressants pour examiner combien de protéines une cellule peut tolérer, peut supporter avant d'avoir un problème de prolifération. Alors, aujourd'hui, on va nous présenter ces travaux. Very much for coming.
Okay. Thank you very much, Christian. Well, I don't understand French, I'm sorry. But my name is Moriya. I'm from Okayama, and today is about constraints of protein expression. I just briefly introduce the place, Okayama, I'm from. This is a map of Japan, you might know. And Tokyo is here, it's Osaka is right here, and Kobe is here. The Okayama is located next to the Kobe. And we have a good research environment, and easy access from Tokyo. We have a Shinkansen bar train. We have also a traditional castle and seafood and foods. And also the famous for denim. It's the original place for the denim where I'm wearing right now. And Okayama is like this. It's kind of square prefecture. The Okayama University is located to the seaside, across the seaside. And then we have several campuses, and we have 11 faculties, and it's a kind of comprehensive university. And the students, like 13 million, that's a bit smaller than University of Quebec, I think. So let me go into the science thing. So the title is Constraint of Protein Expression, and this is the contents of my talk today. So I'm gonna go into the background, conceptual model, the evolutionary principle determining expression level, and then what is the most neutral protein, and toxicity of protein X, poly X sequences. And finally, I would like to introduce the current project called GTO 2.0 project, which is defining design space of protein. That's the final goal. So first of all, I would like to introduce a model of evolutionary principle determining expression level. And it seems to be a complex illustration. So you don't understand right now. I will explain in detail later on this talk. So the background of the talk is from the structure of cell. A cell is a highly complex system composed of thousands of proteins, and we can believe that a eukaryotic cell can perform complex native level functions. So it has a power plant, it has a faculty, border, logistics system, and recycling plant, and also the government nucleus. So these functions are performed through, of course, the proteins encoded on the genome. In the case of Saccharomyces cerevisiae, it has about 6,000 genes located on the chromosome. and which are expressed through the central dogma of molecular biology scheme. So it's transcribed and translated into protein and function connectivity. So in the case of glycolytic system, which is an enzymatic reaction-based system governed by structurally rigid proteins. In the case of cell cycle, which is an interaction-based system mediated by structurally kind of flexible proteins. So, and the expression level of those proteins are very different and tend to correlate with the functional categories. This is the expression of the proteins of growing yeast cells. So expression of some proteins like glycolytic proteins are highly abundant, which express more than 1 million copies per cell. And also, for example, cell cycle genes express just less than 100 copies to more than 10 copies. So the expression levels are quite different, but seems to be somewhat tend to correlate the functional categories. So main topic of this talk is what determines the expression level of that protein, those proteins. Previously, in 2016, Karen et al., which is a Weizmann Institute team, performed experimental analysis, whose name is massively parallel interrogation of effective gene expression. They basically changed the expression of genes by using synthetic promoters ranging the expression level ranging from about 50, 100-fold difference. And they made these strains with different expression level of proteins, and they mix them and measure the fitness expression curve like this. So let me show you one example, which is tab 1. This is a native expression level red line. And if you reduce expression, you will see the reduction of fitness. And if you increase the expression, you will see the reduction of fitness. So basically, most people believe that the expression of the gene is optimized through the evolutionary principle. So what are the evolutionary principles determining the expression level? So we believe that the native expression level is somewhat determines the two evolutionary biases. One is demand, which is created by the functionality of that protein. And if you reduce the expression level by knocking out experiment, you will see the reduction of growth rate. Then you can recognize there's a demand for the increase in expression. The other one is the, we believe, constraints. If you overexpress protein like this, you see the reduction of fitness, and then you can recognize there should be something reducing the expression level. And that in this talk, we correctly call it is constraints, and which is related to the toxicity or cost of protein expression. Okay. So we've analyzed how constraints are happening inside the cell. And in this talk, I just mentioned the expression level that causes a growth defect expression limit. So please remember the expression limit, the meaning of expression limit. And we are measuring, we are trying to understand what is the mechanism to cause constraints. And I summarized some data sets and our own experimental analysis, and we recognized 4 different mechanisms that constrains expression of proteins. This is a fatal protein, which is expressed, transcribed and translated into protein, and some proteins are folding, and then it localized or degraded or interact with other proteins to function, and it has also an activity, which is biologically important activities. And if you overexpress proteins processed through these process, you will see these growth mechanisms to cause growth defect. So let me show you one example, which is pathway modulation. If a protein has activity, of course, protein should have activity for biological function, but if you express some protein, you see the growth defect triggered by the untimely activation or inactivation of protein function. For example, if you express cycle proteins like CDH1 and SIC1, you will see with a strange protein, the cell phenotype that is triggered by the untimely activation or inactivation of cell cycle control. So that is correctly summarized as a pathway modulation. So the enzymatic activity or protein functional activity is increased more than the threshold, we will observe the pathway modulation. So next one is stoichiometry imbalance, which is basically triggered by the misinteraction or the breakdown of the interaction. In this case, this is also the cell cycle protein CDC14, which is a protein phosphatase, but which interact with NET1 to make inactive complex. And then if the CDC14 is released from the complex, it's activated. If you express CDC14 itself, the dosage balance is disrupted, then it causes growth effect. So this is the overexpression of CDC14, you will see the growth effect. But if you overexpress NET1 together, then the balance is recovered, the cell can grow, recover the growth. So this type of growth defect observed by overexpression, we collectively call stoichiometry imbalance. So another reason for the growth defect upon overexpression is resource overload, which is the main topic of the presentation, of this presentation in the first part. So if you express protein, you will see the monopolization, depletion, or competition of resources. For example, transcription resources required, translation resource, folding resource, degradation resource, localization resource are required when you express one protein, and that causes overloading of the resource. And we created another model showing how we can interpret the resource overload, which is a barrel model for resource overload. So these arrows indicates the fate of protein processed through these different processes. And the barrel size indicates the process capacity. And probably the synthesis capacity should be the biggest, and transport capacity of the cell should be smaller. The folding probably that's comparative to the transport or something, but it's smaller than synthesis, and degradation is also smaller than the synthesis capacity. So if you express one protein like this red one, you can predict some process should be overloaded. And in this case, in this red protein case, the folding capacity should be first overloaded because this one has the smallest capacity. So if you express this red protein, folding process should be attacked and disrupted. In the case of, so this process can be considered as a bottleneck process, capacity determined expression limit of this red protein. And another one example is like this red protein. If you express red protein, you can expect transport process should be overloaded. We previously estimated the capacity of transport using Hisao by overloading the membrane transport or nuclear exports or mitochondrial transport. And we've estimated the capacity of mitochondrial transport is about 4% of total protein, and nuclear export is 1%, and the vascular transport is about 0.7% capacity. So, and then the next question is if you remove localization, activation, inactivation, and degradation, and folding constraint, you just remain the transcription and translation process. What you can expect. what you can expect is the overloading the synthesis process. And the point is only unconstrained and harmless protein impose a synthetic burden. Because if it has another the characteristic like transport, then transport should cause the trouble in the first place. So if you remove all the properties, only you observe synthetic constraint. So this kind of synthetic protein production overload is called protein burden, and we've previously estimated it's 15% of total protein. When you overexpress protein in about 50% of total protein, that will cause synthetic overload, viscous overload. Okay, and in that case, you can expect the monopolization, depletion, competition of synthetic transcription, translation resource should occur, right? So let me summarize the first part. And so there is the expression level should be determined by two evolutionary bias, which is demand and constraint. But constraint is not the single dimension. There are several layers of constraints determining the expression limit of a protein, and the least or strongest constraint determines the expression limit. But if you remove, for example, activity or localization, you can increase the expression limit to ultimately the expression limit overloading synthesis, which is previously estimated about 15%. Okay. And then the point is only unconstrained and harmless protein can cause protein burden. So what is the specific research on protein burden? We actually, including our three groups, including ours, used fluorescent proteins because it can consider as non-functional, no interaction partner inside each cell, and not localized to anywhere, and fold itself, and not actively degraded. So those protein doesn't have any characteristics to cause other troubles than the protein burden. And there are three groups, including our group, performed a protein burden analysis. So Kafri and Barkai, it's an Israel group, is using M-Cherry, and Farkas, it's group using Venus, and our group is using EGFP. So they are different fluorescent protein, but we believe that they have no functionality or no localization or no interaction patterns. And but the problem is actually the data set, the result is somewhat different. And the main problem is that if you express Venus and EGFP, we observe cell elongation phenotype, and also we observe the overloading of the chaperone system, like HSP70 aggregation inside of the cell. So our prediction was, first prediction of ours was wrong, because these proteins require somewhat folding resources. So these proteins are not good for the model protein to analyze the protein burden because it requires the folding resources. Then we have to use better protein without these folding programs to understand the synthetic burden, right? So we changed the target protein into MOX-GFP, which has improved folding property without cysteines. So we no longer observe cell elongation phenotype and no longer observe the aggregation of HSP70. So now we believe that this protein is a good model protein. However, we recently found that this one is not the best protein, because if we put a mutation on the fluorophore to change non-fluorescent fluorescent protein, the protein expression level, the expression limit is increased about threefold. This has 15% of total protein is the expression limit, but this protein has 43% expression limit, which is, we believe, the record to be expressed in E cell and probably in E. coli cell as well. So, and we also observed some depletion, ribosome process reduction, production reduction, and then N-source depletion. I'll show you later. Anyway, surprisingly, activity or fluorescent something caused the trouble for when we overexpress EGFP or fluorescent proteins. So now activity is also can become a constraint of fluorescent protein. And then when you remove that activity, the expression limit is increased up to 43% of total protein. Okay, what do we learn from that experiment? Before go into the detail, I'm going to explain about the experimental system. We call it is a genetic taggable, which can express protein up to the protein, growth defect limit. In this case, protein burden limit. We use 2 micron plasmid with due to the selection marker. By using this selection marker, we can increase the plasmid copy number of the cell in leucine deficient medium up to more than 100 copies, because LU2D is a leucine synthesizing enzyme gene, but expression level is really low. So the higher copy number becomes a bias to increase the plasmid copy number. And also we put a target gene on the same plasmid. So the plasmid copy number is increased, the protein expression should be increased. And then if it reaches to the expression limit, the plasmid copy number should be less than the limit. So determines the taggable between the target gene expression and LU2D nutrient requirement, determines the expression limit of the, expression level of the cell of the target protein, which is related to the expression limit of the target protein. We use TDH3 promoter, which is the strongest promoter, so that we can express really, really high level inside the target protein inside the cell. So the discovery is like this. This is my notebook in 2020. When we put a mutation on the chromophore, like a tyrosine 66 is changed to the glycine, you will see the increase in the protein level up to threefold. So we try to look at the systematically try to look at the effect of fluorescence. And this is the systematical analysis. We use EGFP, superfolder GFP, MOX GFP, and mCherry. And when you put the mutation, non-fluorescent mutation, the protein level expression limit is all increased like this. And we also measure the growth rate using the calculation from the growth curve. And the growth is somewhat different, but mutation cannot change the growth rate. So the protein is expressed, but the growth rate is not so much changed. So we need to try to make which one is the most toxic and which one is not toxic by using these data. To calculate the toxicity, we introduced a neutrality index, by which we calculate the just multiply the percent max growth rate and max protein level. For example, the protein can be expressed inside of the about 100% of protein. Of course, it's impossible, but hypothetically, if you express a protein with 100% of total protein, and even though the cell can grow 100% of growth rate, you can tell the protein is completely neutral. Of course, it's impossible. And if you overexpress protein, but the protein level is really low and the growth rate is really low, then you can tell the protein is really toxic. So to evaluate this kind of relationship, we calculate the neutrality index by multiplying growth rate and protein level. And it turned out that the non-fluorescent protein, MOX GFP, is the least toxic most neutral protein. So we decided to use this protein as to understand the physiology of protein burden. But before I go into the detail, I just want to show some comparison between the previous data. So as I said, the M-cherry is also used as a model protein to analyze the protein burden. And but the point is in this research, they have observed the cell size increase. The cell is elongated. And it's reported that's the response of the cell itself to the protein burden, because they want to increase the protein synthesis capacity in response to the protein burden, then it will cause the cell size increase. However, when you mutate, we found that when you mutate the fluorescent core, fluor4, you don't see any cell size increase like this. So eventually overexpression causes the cell size increase, quite enlarged. But if you mutate in the fluorescent core, you don't see the increase in the cell size anymore. And also the protein level, or I would say the neutrality is increased. So the protein is highly expressed, but the cell size elongation or cell size increase is not a general phenomenon. Only observed excess fluorescent imagery. We don't know why it happens so far, but this is not at least the response for the protein synthesis overload. Okay. So we performed a transcriptome analysis in MOX-YG overexpression and MOX fluorescent protein overexpression and non-fluorescent protein overexpression, and compared the physiological response. The main finding is that ribosomal biosynthesis, which is previously believed that increased upon protein burden or protein synthesis stress, but it's actually conversely it's reduced. So the protein ribosome production is surprisingly reduced. And we also observed the N-source depletion as the transporter for amino acid or nutrient, some nitrogen source is increased when MOX-YG is overexpressed, but not MOX overexpression. So we finally observed the depletion competition of synthetic transcription translation resources. And we also performed the proteome analysis. And in this case, we introduced one additional control gene, which is frame shift mutant, that is transcribed but not translated. So it's only overall the transcription process, but not translation process. And in this case, we didn't observe any growth defect. Okay. So the translational defect is most important part of the protein burden. And this is the measurement of total protein compared to the vector. Important finding is that the total protein amount is not different between the overexpression and the vector control. And the total amount, the other proteins like than expressed MOX-YG is reduced about half, about 50% of total protein. So, and then if you describe that situation into the proteome map, you see about half of the protein is covered by the MOX-YG, and other proteins are basically reduced. So, and then we try to look at the difference in the context, the composition of proteins. And we found that the ribosome biogenesis genes and ribosome proteins are basically reduced, and glycolytic proteins are also reduced. However, the oxidative phosphorylation proteins, like mitochondrial proteins, are increased. So this is a summary of the proteome change. And previously, the nutrient limitation activates a TORC pathway. Inactivate a TORC pathway, which will change the proteome composition similar to that situation. So we compared the overlap between the TOC pathway, the protein changed in TOC pathway inactivation and the MOX gene of expression. We observed a significant overlap between the up gene and down gene. So namely, probably overexpressing non-harmful protein will cause the nutrient limitation that will reduce the expression of ribosome proteins and also increase the proteins involved in oxidative stress. Right. So massive expression of protein and source depletion, TOC inactivation, and maybe cell is going to the economy mode. And we still have several unsolved questions. So if you look at the cells under the electron microscopy, we see this is the nucleus and this is the nucleolus. So we see apparent defect in the nucleolus composition. And we also observe the reduction of cytoplasmic density because ribosome is probably reduced in this situation. And we also observe the top speed of the growth is reduced. Basically, it will retard the recovery from the initial state to the log phase. So there is a delay of the growth. But this growth retardation is basically coming from the retardation in the first top speed growth. So the top speed is somewhat growth in MOX gene of expression than the vector control. And then we also observe the growth defect at high temperature. Like if you grow it at high temperature, the cell cannot grow to the final cell density is highly reduced compared to the 30-degree or vector control. And we don't know why it happens. We have to solve these issues. But anyway, in the first part, I would like to introduce about this model. And so the protein expression levels are determined by demand and constraints. And the constraints are created hierarchical structure. The most limiting one determines the expression limit. And when constraints are added or removed, the expression level is increased or decreased. And finally, ultimately, the protein burden of synthesis, the burden of synthesis is the constraint for all proteins. Its final constraint is about 40% of total protein. Okay, this is the first topic. So the next topic is, this is one first and the second topic. And the third topic is what toxicity of poly-X sequences. So if this kind of demand of the constraints is existing, we can analyze, or our experimental system, using our experimental system, we can analyze the constraints by using different proteins. Okay. So we first, very simple question. So what are amino acid sequence which can be constrained or unconstrained? And we analyze very simple model protein or model amino acid sequence, which is poly-X, poly-10X sequence. So that is representative of amino acid sequence. If you try to analyze 10 amino acid from 20 amino acid, different amino acid, it has 100 billion, 100 trillion combinations. So we cannot test everything. So we just focus on very simple model sequence, which is we call poly-10. So which is a sequence of 10 identical amino acids. In this case, we call it poly-L3. This one is poly-10, poly-D6. But we focus on poly-10, poly-X10. So to try to analyze the toxicity of the poly-10 sequence, we add the amino acid poly-10, poly-X10 sequence at the C terminal of EGFP. And we created this kind of construct for 20 different amino acids. And we measured expression limit and growth effect using the same scheme as I mentioned previously. And this is a result of the toxicity of amino acid sequence, 10 amino acid sequence. So basically, hydrophobic amino acids or large amino acids are quite toxic when it's 10 conjectively aligned. And these positively charged amino acids are intermediate, and negatively charged amino acids or hydrophilic amino acids are kind of neutral. And the interesting finding is if you put poly-E glutamate, you see the increase in the growth rate and expression level than EGFP itself. So this sequence can somewhat reduce the toxicity of EGFP, which is probably avoiding the misfolding or something. But if this trend is actually functioning as an evolutionary bias for the amino acid sequence composition in proteome, we could see the same kind of trend. So we look at the proteome of each cell and try to find this easy actually bioinformatics analysis to try to find how long the amino acids are consecutively aligned. And in the case of, for example, poly-D, you see 43 D as consecutively aligned on a sequence. And but however, if you see W or M, you don't see anything more than 3. So it's strictly constrained to not to be aligned in a same way. Okay. And the trend actually is quite correlated. So this actually shows a poly-X max, how many amino acids are aligned in the same consecutive way. And this axis shows the experimental data. We measured max growth rate and EGFP overexpression experiment. And then we see a good correlation between experimental and the proteome composition. So that means probably our findings, some amino acid sequences are strictly constrained and some amino acids are not so much constrained in evolution. Okay. Well, I will read. The toxicity neutralities of amino acid can serve as an evolutionary bias in protein sequence composition. Okay. So let me go to the final part, which is ongoing project. We call it GITO 2.0 project. So this is the model I showed in the previous talk, and the evolutionary principle determining expression level. And if you split this model to a second dimension space like this, it's a bit complicated. But if you split this model into the second dimension model, you can tell design space of a protein. So this axis, this line axis shows the expression of a protein, and the blue area is the accepted expression area. And this part is actually over here. So native expression level is determined by the requirement, demand structure, and constraint structure. And there are many different constraints constraining the expression of a protein, which we can call a constraint structure, which determines the upper limit of the protein. So if you, for example, remove the enzymatic activity, then you can increase the expression of a protein because this bias is no longer observed. And then you see aggregation would be the second constraint. So the protein expression level or protein function can be performed inside of this demand, outside of the demand structure and inside of the constraint structure. So the final goal of ours is to determine the whole try to understand the whole structure of a constraint. To do that, what we need is to understand what kind of constraints exist. So the goal is the drawing whole design space of proteins, and that could be the design space of the cell. But to do that, we have to understand, or we have to know what are the constraints. We have to understand everything. So to do that, we started a project that tried to measure the expression limit of all protein of each cell. So this is basically performed by the Shotaro Namba, who is a former PhD student. Now he's a postdoc, and he's over there. And then we created this system. We just add the difference from the original GTO is we use WTC promoter, whose activity is changed by the amount of drugs. So we can control the expression by using tetracycline, TATC. And then we can measure the expression level of protein by fusing MOX-GFP, which is kind of least toxic fluorescent protein. So we can perform wide range and ultimate high-level expression, and we can measure the expression limit. And for the proof of concept, we cloned about 80 proteins located on chromosome 1. Chromosome 1 encodes about 96 sort of protein, and 80 can be cloned because the other one is somewhat, the structure is somewhat broken at the end of the chromosome. But this can be considered as a random pickup of the proteins. So no functional bias is not observed. So, and we measured growth rate and expression level, and we also performed some microscopic analysis and also the viability measurement. And then we did data analysis to calculate, to extract what protein is toxic and what protein is not toxic. And I'm not going into the detail, but I just want to briefly summarize the result of this part first. So the aim of this analysis to make the protein or express protein until it causes the growth defect. And previously, it's impossible because the power of the induction is not strong enough. But by combining the strongest promoter and the GTO system, we can now reach to the expression limit of every protein. So this is the result. We measured growth rate and the calculated max growth rate, and the strongest expression condition we observe, most of them reach to the expression limit. That causes growth effect. That's the red point. And we also try to measure or try to calculate neutrality index by using the growth rate and expression limit. So this is the second dimension graph, which x-axis shows the growth rate, and y-axis shows the growth rate, and x-axis shows the expression. So expression, growth rate curve. From these curves, we try to get the neutrality index, which is multiplying the max growth rate, the max expression level. So by using that, we reduce the multidimensional data to one neutrality index, okay, like this. And then now we got the toxicity index for proteins, or neutrality index for proteins. Then we can compare or perform association study with other parameters like expression level, like length, like amino acid composition, localization, and some other parameters. And we surprisingly found that the most highly correlated parameters is PLDTT of AlphaFold prediction. So a PLDTT prediction is like if protein is very bluish, the PLDTT is really high. Then if the protein is orangish, the PLDTT is low. So basically that is correlated. That is a representation of the confidence of prediction. So basically, the highly confident proteins are neutral. So those are basically the enzymes expressed in high level, and it has a rigid structure and evolutionary concept. They are actually least toxic proteins. And also, these proteins with disordered regions, the structure prediction is impossible because they are disordered, but those proteins are highly toxic. And another interesting finding is that on chromosome 1, several young genes, young proteins, which might not be functional, which might be encoded, it happened to be encoded, but it's not functional DBS protein. And we also analyzed those young or non-functional proteins. They are actually quite yellowish. And then their proteins are quite intermediate. They are not too toxic, and they are not too non-toxic. So we believe that the evolutionary rule of neutrality toxicity might be revealed by this analysis. So the newborn genes are intermediate, but to become toxic or non-toxic, they should be evolved. Okay. So another experimental the data we believe it's quite interesting is the microscopic observation. So when we overexpress proteins in really, really high level, we observe very strange visual structure, intracellular structures emerged through GITO 2.0 analysis. These are the examples, so which are not observed previously. Of course, it's quite artificial, but we can tell probably this is a potential to construct a structure, intracellular structure, might be revealed by this analysis. For example, if you express FAN12, we observe very sticky cytoplasmic conditions. And if we overexpress PEX22, which is a putative peroxisomal membrane protein, we will observe very strange structure, which we call gorgon. That is a tubular structure coming from the lipid droplet. So this is a membrane binding protein. So if you overexpress this protein, maybe it will convert some structure of organella into this bizarre structure. But we believe that this kind of thing is a potential of a cell to create sort of new organelle. So this structure is basically triggered by the N-terminal region of the PEX12. And if you mutate these charged amino acid, the gorgon is no longer observed. Okay. So this is the final presentation, final slide. So the aim of GITO 2.0 project is there are two aims. So first one is understanding cell structure, full structure of biological constraint to enable flexible cellular control and relational protein design. So in this project, we want to make a model based on expression limit and measurement and phenotype profiling. So the concept is that from the amino acid, we can now probably using some AI-based models, we can predict the complex formation and localization. This part is now we can do it. But what happens inside the cell if you overexpress those proteins or just expressing proteins inside the cell is not predictable. So we want to make this kind of constraint structure by measuring the cell state, extensively measure cell state upon overexpression of proteins with some properties, and to construct this kind of, try to make this kind of whole structure model. And the other one is discovering possible cellular state beyond this limit. So that if you express protein beyond the limit, the cell will go to the different state, or probably catastrophic or evolution. So one example is PEX22 expression, which will cause the probably new functionality or new structure. And we also observe several mutants that overcome your expression. Like this, if you express, in this case, GEM1, the cell shows very strange aggregation and causes growth defect. But some mutants, adapting mutations can solve that issue and then can grow faster than the wild type. So that is probably the outside of the constraint that can evolve the cell into the different state. So this is the end of my talk. I would like to thank my students perform these kind of experiments. So FP toxicity is performed by Yuri Fujita, and Poly 10 project is performed by Murase Yukihiro, and G2 2.0 is performed by Shotaro Namba, who is over there. Thank you very much.